So we're here with Dr Ameur, who's recently got an adjunct appointment with the School of Public Health and Preventive Medicine at Monash. So Adam, why don't you tell us a bit about yourself and your work?
Yes, so I work at the National Genomics Infrastructure in Sweden at Uppsala University. And I've been for a long time working with next generation DNA sequencing and also with the bioinformatics. So recently I've been involved in a big project in Sweden where we have sequenced the genomes of 1,000 Swedish individuals with the aim to construct a reference database for genetic variants. So with this genetic variant database, we can sort of look at the background frequencies in the Swedish population and use it as controls for many different types of studies.
Yeah, I think it's a really important thing that the field needs at the moment is this basic characterization of a reference population so we can understand what the genomes of a population look like. And so our work here has been involved in a very similar project in Australia called the Medical Genome Reference Bank, where we've now done whole genome sequencing on 3,000 healthy elderly individuals, and 2,000 of them have come from the Esprit Healthy Aging Biobank at Monash. And so we've now generated a huge amount of data in a similar way to what you guys have done in Sweden. And we have now the huge task of kind of cataloging all of that variation that we can now measure with this technology and making it available to the wider research community so they can utilize that data. So why don't you tell us a bit about how that making the data available in Sweden has happened and some of the benefits that that's giving the research community?
Yeah, so what we did was to put up this information on a website. Of course, it's difficult also with genetic information. We cannot, put information about each individual available on the website, but instead we have generated frequencies for the general population. And with these frequencies, it's something that everyone can go in and just look at specific variants and see, okay, what is the prevalence of this in the population? And if you then are looking at a patient, for example, with a specific disease, where you suspect that you have one causative mutation, then you can see, okay, what is the frequency of this mutation in the population? And that has been very important for the clinicians.
And so if we take a step backwards, whole genome sequencing is now the ability to read every letter in the genetic code almost, not quite every, but it amounts to about 3 billion letters or 6 billion letters with the two chromosomes in each human genome. And then the differences between each individual is also massive amount of variation. So what were the kind of numbers that you picked up in terms of the number of coding changes or letter changes that you were picking up per individual and across the whole data set?
All right, so for each individual, we in fact find millions. I think it's about 4 million variants that are different than from the sort of reference genome. And many of these are then in coding regions that could have an effect on the protein. So that's quite massive amount of data, and we also found many novel genetic variants that seems to be specific for the Swedish population.
Yeah, and I think every time we sequence a new genome, we're discovering thousands or hundreds of thousands of new variants that have never been seen before in another sample.
Yeah, and that's fascinating.
Yeah, it's just staggering how much variability is out there. So you mentioned that the paradigm that we're currently operating in is comparing these sequence reads versus the reference sequence, which is an artificial code that is supposed to represent a human genome, but in fact it's been compiled from multiple people's DNA, and it's been kind of improved and built upon for many years. And there's the possibility that we might move away from that paradigm, not being so reliant on the reference sequence. But maybe you could tell us a bit about your work that has been really getting into the structure of that reference sequence and how we can potentially improve that sequence.
Yes, so in fact for two of these individuals in the project, we have been interrogating them with other technologies to generate new reference sequences for the Swedish individuals. And what we have seen is, in fact, that there are plenty of gaps in the reference that we're using now. So we are aiming to fill in some of these gaps and perhaps generate more information about the human genome. So the human genome is still not complete, it seems.
Yeah, and I know that you found sequences in your samples that weren't even in the reference sequence.
Yeah.
And so, yeah, I think we're still learning about what a genome really looks like
Yeah,
in
we
one
are.
individual
Yeah.
from the next.